Welcome back to Gyanvarsha. Today we are today we will discuss about human metapneumovirus. So why this topic is very important? This topic is important because this is a very current topic nowadays in news channel in each and everywhere we are seeing this human metapneumovirus. If people are saying again lockdown will be there, these type of people are saying, but we need to study about this human metapneumovirus because for our exam point of view, like they can ask maybe some question. Some one of my junior, he was telling that he wrote this ETREC exam, and in that exam in that this human metapneumovirus that related questions came. So we'll understand what is human metapneumovirus. Okay, so in this topic we will cover some of the basic things like it was when it was first discovered, its discovery we will understand, how it was isolated we will understand, its origin we will discuss. Okay, then its molecular organization we will discuss, molecular organization we will discuss. Then we will discuss its pathogenesis also, or is there any clinical treatment, or any treatment is there or not for this virus. We will discuss these things. Okay, so first we'll move, what is this HMPV virus? HMPV, what is this HMPV? Human meta human meta pneumovirus, HMPV. It is a viral pathogen. It is a viral pathogen. It was first discovered in 2001 in the Netherlands. It was first discovered in the 2001 in the Netherlands. Okay, you understood this point? How it was isolated? So it was isolated. So this virus was isolated from the pediatric patients. Isolated from the, you know what is pediatric patient? Pediatric patient. Pediatric patient. Pediatric patient means the like pediatric means healer of children. Healer of children. Okay, so it was isolated. So this virus was isolated from a pediatric patient who had symptoms similar to those of HRSV infection. Again, I am repeating. It was isolated from the pediatric patient who had symptoms similar to the HRSV infection. What is this HRSV infection? You will further get to know in further slides, but here I am telling you the full definition of this human respiratory syncytial virus. Syncytial virus. Human respiratory syncytial syn, syncytial virus. Okay, so I hope this much part is clear. HMPV is a viral pathogen. It was first discovered in the Netherlands when the virus was first isolated from a pediatric patient who had symptoms similar to those of HRSV infection. Okay, so we'll first understand like what like I hope you know about this kingdom, phylum. This we read, na, genus, order, species. So see, this I'm telling you, the order was mono mononegavirales. Order was this one. Okay, this order, it this it has a family. Family is para myxoviridae. Okay, this is the family. This is what a family. Now this family, oh wait, this is a family. Okay, now what happened? This is a family. Now this family is further divided into two subfamily. One is this para para mixo para mixo virinei virinei. Okay. Second family was pneumo virinei. See, the order is the order is mononegavirales. Then the family is paramyxoviridae. Then this family was further divided into two subfamilies, paramyxovirinae and pneumovirinae. Okay. Now this this pneumo this pneumo family is further subdivided, okay, into two genera, pneumovirus and metapneumovirus. This HRSV is placed in this HRSV is placed in the genus pneumovirus. HRSV is placed under this pneumovirus, while the Hmpv is placed under the metapneumovirus. Hmpv is placed under this metapneumovirus. I hope you are understanding what I am trying to say. The order is mononegavirals. Then if we'll come to order to family, the family is paramyxoviridae. The family is divided into subfamily, paramyxoviridae or pneumovirinae. This pneumovirinae is further classified into pneumovirus and metapneumovirus. So HRSV is coming in pneumovirus, while the Hmpv is placed under metapneumovirus. Okay. So you know what happened when this whole genome sequence analysis was done. I don't know the here I am writing, when the whole genome sequence analysis was done. So this Hmpv exists as two genotype, A and B. Again I am repeating, when this whole genome analysis was done, whole genome analysis was done, this Hmpv exists as two genotype A and B. Okay. Then they further classified into subgroups like this. A was classified into A1 and A2, and B was classified as B1 and B2. You understood? Again I am repeating, bachcho. See, this order is mononegavirals. Family is paramyxoviridae. This family was further subclassified into paramyxoviridae and pneumovirinae. This pneumovirinae is further classified into pneumovirus and metapneumovirus. HRSV belongs to pneumovirus. It's placed under pneumovirus, while the Hmpv is placed under metapneumovirus. Then when the whole genome analysis was done, this Hmpv exists as two genotype A and B. This A was further subdivided into subgroup A1 and A2, while the B divided into two subgroup B1 and B2. Okay. Further then A2 also again subdivided A2A, A2B. This A2 again further subdivided A2A and A2B. So you don't need to go in this much detail. A2B, small b. Okay. So here I hope this much part you understood. So we'll go to molecular virology. What is the molecular virology of this virus? So this is a Hmpv virus. Okay. So it is first of all, it is polymorphic in nature. Not polymorphic, pleomorphic in nature. Pleomorphic in nature. Pleomorphic in nature. Okay. Its size varies from, this virus sizes varies from 150 nanometer to 600 nanometer. Its size varies from 150 nanometer to 600 nanometer. One thing I want to tell you, when we are talking about its genome, Hmpv genome, it's a negative. It's very important point. Its genome is negative sense single stranded, negative sense single stranded RNA, negative sense single stranded RNA, and it contains 8 genes, and it contains 8 genes which code for 9 proteins. 8 genes code for 9 proteins. 8 genes code for 9 protein. Okay, again I am repeating bachon, this Hmpv genome is negative sense single stranded RNA. It contains 8 genes which codes for 9 protein. Okay, so we'll move further. What are the order of genes? It is very important to understand. See, I will tell you what's the difference between this RSV and Hmpv. See, if you see this Hmpv, this first figure, 3 prime to 5 prime direction, this N, P, M, see there are 8, N, P, M, F, M2, SH, GL. So, N stands for nucleoprotein. N stands for nucleoprotein. Okay. P stand for phosphoprotein. While M stand for matrix protein. F stand for fusion glycoprotein. Fusion glycoprotein. Then M2 stand for, it is a this is a transcription factor. Putative transcription factor. Okay. Then see, this is M2, there is 2, M2 1 and M2 2. Okay. Then next SH is small hydrophobic hydrophobic glycoprotein, small hydrophobic glycoprotein. Okay, then G stand for attachment glycoprotein. It is also a glycoprotein, but it is attachment glycoprotein, while SH is small hydrophobic glycoprotein. And last L stand for, it is what? A viral polymerase. It is what? Viral polymerase. So these are 8 genes which code for 9 proteins. I am saying this N, P, M, F, M2, SH, G and L. Okay. So I can tell you how these are genes. I can tell you how these 9 genes will 9 proteins will be. See, N, P, M, F. M2 will further classified into M21 and M22. M21 and M22 protein. Okay. So this N protein, P protein, M protein, F protein, M21 protein, M22 protein, SH protein, G protein and L protein. I hope you are understanding this much. Okay, this M21 protein is a transcription factor, while M22 protein will help in RNA synthesis regulatory factor. M22 protein will help in RNA synthesis regulatory factor, while M21, what it will do? It is a putative transcription factor. Okay. So now see the structure of this. See here one more difference. See, this is what a Hmpv genome organization, while this second figure represent the genome organization of HRSV. So here you can see the difference. One very important difference you can see here is, I'll show you with different color. This blue color. You can show here this NS1 and NS2. What is this NS1 and NS2? These are two non-structural protein. What are these? These are two non-structural protein. So it may be, so it may be the reason for the difference in level of host innate immune response. So it may be the difference, this HMPV and HRSV, because in HRSV this NS1 and NS2 are present, while in HMPV this is absent. So it may be the reason for the difference in level of host innate immune response observed during this HMPV and HRSV infection. Okay, so this may be the reason, because of these two non-structural proteins are present. So what these non-structural protein will do? It it have been identified as potent. You can say this. How should I tell you that it has to be identified as potent multifunctional antagonist of IFN signaling, interferon signaling. Okay. If you are not understanding, leave this, but just remember these are two non-structural proteins. So next further we'll move. So see now the structure. So this is very important. See this structure. This is what? This is, see the RNA core. This RNA core is surrounded by, I'll use some other color pen. Uh, okay, black color I am using. Okay. See, this RNA core is surrounded by M protein, which is surrounded by M protein. If you see, this is what a M protein, this blue you are seeing, this is surrounded by, this RNA core is surrounded by M protein, as well as covered by a lipid envelope. This is what a lipid envelope. So this lipid envelope, listen to me, this lipid envelope contains F, SH and G. F, SH and G. What is this? Lipid envelope. This lipid envelope, what? It contains three surface glycoprotein F, SH and G. Okay. The you can see in the form of what? Spikes. These are what? Spikes. Can you see this? Spikes. These are what? Spikes. See the spikes here, this spike, red color spikes, this star type spikes. So in the form of spikes. Okay. Okay. So now further we'll move. We'll understand how this is working. So it's very important. See, we understood what is N, what is P, what an N protein. So we'll understand what is this mechanism. So understand this thing. See, what will happen with the help of, I'm writing here, with the help of G and F protein, with the help of G and F protein, this virus HMPV attaches and fuses to heparin sulfate, to the heparin sulfate. Okay, heparin sulfate receptor. Okay, when after the fusion, after attachment and fusion, the viral the after fusion, the viral new the viral nucleocapsid enter into the enter into the cytoplasm of the host cell. Cytoplasm of the host cell. In simple term, I am telling you, see, suppose this is what a virus. Okay. This is what a virus. This is a human cell. Okay. So what it will do, this G and F protein is attach and fuse it with this heparin sulfate receptor. Then after fusion, this it has a nucleocapsid. So this nucleocapsid will enter into the cytoplasm of the cell. Okay. After that, what will happen? The newly synthesized viral genome assemble with the listen, after entering the nucleocapsid into the host cell, into the host cell of the cytoplasm, what will happen? This newly synthesized viral genome, newly newly synthesized viral genome assemble with other proteins like P, N, L and M2 protein. Newly synthesized viral genome assembled with other viral proteins like P, N, L and M2 proteins and move towards the host cell membrane and move towards the host cell membrane. Okay, from there this viron now bursts out of the cell, and from there what will happen? This viral now bursts out of the cell. Okay, with the FSH and G protein. What are the FSH and G protein? These are an lipid envelope, with lipid envelope. Lipid envelope, now you are getting what are lipid envelope proteins? FSH and G protein. Okay, exposed on the outer side of the membrane, which has to be exposed on the outer side of the, see, I didn't find any diagram, that's why I'm telling you in that like in that point. Okay, after that what will happen? Here I am writing this. After that what will happen? The pre protein, the P protein, after that what will happen? The P protein, which is phosphoprotein, the P protein act as cofactor to stabilize the L protein. Why P protein is needed? To stabilize the L protein, to stabilize the L protein. Okay, L protein is what? It will do viral polymerase. So it will P protein act as a cofactor and it will stabilize the L protein. So what will happen? Allowing the formation of virus RNP, ribonucleoprotein complex, RNP, ribonucleoprotein complex. Okay, formation of RNP complex. Okay, then M protein, what M protein will do? M protein will play a very important role in virus assembly. M protein is matrix protein. M protein, what it will do? It will play a very important role in virus assembly and budding by interacting with RNP complex. See, it's very easy. Again, I will repeat. Don't worry. RNP is what? Ribonucleoprotein complex. Then the N protein encapsulate the viral genome, and what N protein will do? See, N protein is nucleoprotein. So N protein will protect it from nuclease activity. N protein protect the protect it from nuclease activity. Okay, and you remembered, see, at first I am again repeating these steps. What will happen? G and F protein, what it will do? They will attach to the this Hmpv virus, attach and fuse to the heparin sulfate receptor. After fusion, the viral nucleocapsid enter wait, I'll change the color of the pen. After fusion, this viral nucleocapsid enter into the cytoplasm of the host cell. After that, this newly synthesized viral genome assemble with other viral proteins like P, N, L and M2. they move towards the host cell of the membrane. They move towards the host cell membrane. Then this viral now bud out of the cell with other with lipid envelope. What are the lipid envelope protein? This F, SH and G protein. Then what will happen after that? The P protein, phosphoprotein act as a cofactor to stabilize the L protein. Then the formation of RNP complex, this ribonucleoprotein complex. Then this M protein will come. It will help in virus assembly and budding by interacting with RNP complex. Then this N protein, it will protect the from nuclease activity, like nuclease will not degrade the this ribonucleoprotein. Okay, so means it will not degrade this RNA. Okay, so N protein will protect it from nuclease activity. Then one more protein remaining, if you remember, this M21 protein. So what it will do, this M2, not M21, M22 protein, what it will do, M22 protein, what it will do, it will decrease the host innate immunity. It will decreasing the host innate immunity. Host innate immunity. I hope you understood what I am trying to say. Students, again I am repeating one more time. It's very important. It is very easy. Nothing is there. There are 8 genes which code for 9 proteins. See, first G and F protein. What it will do? It will attach and fuses with the heparin sulfate receptor. Okay, with the help of that, with the help of G and F protein, Hmpv attach and fuse with the heparin sulfate receptor. Then after fusion, this viral nucleocapsid enter into the whole cytoplasm of the host cell. Then this newly synthesized viral genome assemble with other viral protein P, N, L and M2. Then this this will move towards the host cell membrane. Okay, this newly synthesized viral will move towards the host cell membrane. Then it will bud out of the cell with viral with the lipid envelope. Lipid envelope proteins are F, SH and G. Then the P protein act as a cofactor to stabilize the L protein. Then the formation of RNP complex. Then M protein will come to form the virus assembly and budding by interacting with RNP complex. Then N protein will protect it from nucleus activity, while the last protein M22 protein, it will decrease the host innate immunity. Okay, so I hope this much part is very this part is clear for you. So now the main question arises. So one question, not main question, one question arises, when this happen, when mostly these when we can see this type of when we can see its symptoms. So mostly we can see spring and winter months. Spring and winter months. Winter months. Okay, so January to March in northern hemisphere, while June to July in southern hemisphere. June to July, June to July in southern hemisphere. Okay, maybe they can ask. So see, I am covering most important points. So next we'll move. Okay, one more important question. So maybe they can ask how we can detect this, like how we can see. So if you remember, corona virus also um, we diagnosed it by RT-PCR. So this is also, so because of its slow growth of this virus in the cell culture, molecular methods such as RT-PCR are preferred diagnostic method for detecting Hmpv. What I am saying, RT-PCR, reverse transcriptase. Okay, reverse transcriptase polymerase chain reaction is the preferred diagnostic method for detecting Hmpv. Why? Because it has very slow growth in the cell culture. This virus has very slow growth. Okay. So now we'll move forward to the pathogenesis of Hmpv infection. So I'm explaining you in very easy way. Listen carefully so you will understand. See, first I will explain this. Okay. See, suppose virus trapped by toll-like receptor, either it's macrophage or dendritic cell. Dendritic also in blood stream. Okay. Virus got trapped by toll-like receptor. Then what will happen? Several adapter molecules like M MYD88 MYD88 and TRIF. These are what? Where is my pen? Ah, these are adapter molecules. What it will? They will. These adapter molecules. See, first virus got trapped by dendritic cell or macrophages. Then this adapter molecule will activate. Okay, this adapter molecules like MYD mid 88 and TRIF got activated, and which in turn activates the NF kappa B. How NF kappa B will activate? It activated by IKK. It will activate by IKK. So which NF, when IKK will activate NF kappa B, okay, then NF kappa B will bind and do its own functioning. While on the other hand, this virus, endocytosis of virus, this viral RNA will come inside of the host cell. Then this RLR, cytoplasmic RIG1, I am writing this its full form, RIG1 like receptor, like receptor. It got activated, which in turn activates NF kappa B. How NF kappa B will activate? NF kappa B will activate first MABS will activate, MABS will activate IKK, IKK will activate NF kappa B. Okay. MABS is a mitochondrial antiviral signaling system. Mitochondrial antiviral signaling system. Signaling protein. Signaling, sorry, it's a protein. So MABS is signaling protein. So MABS will activate IKK, which in turn activates NF kappa B. Okay. Or while the other hand, this IRF, interferon regulatory factor 3 and interferon regulatory factor 7 will activate. They both activate and act as a transcription factor and binds to the nucleus, go to the nucleus. Then it will release interleukins like interleukin 2, interleukin 4, interleukin 8, interferon alpha, beta, gamma. Okay. So again I am repeating what's the pathogenesis of Hmpv infection. Listen very carefully, you will understand. See, this virus trapped by TLR. TLR is what? Toll-like receptor. Either they are toll-like receptors are present on macrophage or dendritic cell. Then once it will get trapped, this adapter molecule like mid 88 and TRIF will activate. What it will do? It will activate NF kappa B. How it will activate? It will activate IKK. Okay. This IKK will activate and it activates the NF kappa B. NF kappa B act as a transcription factor and go to the nucleus and release several interleukins and interferons. While on the other hand, if the endocytosis of virus will happen, then viral RNA will come in the cytoplasm of the host cell. What it will do? This RLR, RIG1 like receptor, it got activated. It activates the MAVS protein, which is mitochondrial antiviral signaling protein. It will activate the NF kappa B. How it will activate? It activates the first IKK. IKK will activate the NF kappa B protein. Okay. Then NF kappa B again go to the nucleus. While on the other hand, RLR can activate IRF3 and 7. Okay. RIF3 and 7 goes act as a transcription factor, goes to the nucleus, and then it will release interleukin and interferon. So this is a pathogenesis of the Hmpv. So I hope this pathogenesis you understood very easily, and it's very easy. So next we'll move forward. So see, there are some control treatment strategies, but there is no treatment. I don't think so. There is no any treatment which is commercially available, any vaccine or anything which is commercially available. But some very famous are some antivirals. So its product is ribavirin. So this for this human like tissue culture assay used and human model also used. Okay, for that antibodies also monoclonal antibody produced, fusion inhibitors like inhibitory peptide, you know, RNA interference also, RNA interferon also used as a control strategy, and siRNA is the product of this. Antiviral is a control strategy, ribavirin is a product, RNA interference also used as a control strategy, and is the product is siRNA. Okay, so there are a lot of I hope this much. One is epitope vaccine also, epitope vaccine. So for that T lymphocyte epitope vaccine is the product. Okay, then inactivated vaccine also used as a control strategy. So product is heat inactivated vaccine. Okay, vaccine also. Chimeric I taught you in monoclonal antibody what is chimeric vaccines. So antigen hmpv antigen on para-influenza vaccine is the product. Para-influenza vaccine. Okay, one more vaccine, subunit vaccine also they used as a control strategy, and the product was hmpv f subunit vaccine. F subunit vaccine. Okay, I hope that much. Live attenuated also they used as a control strategy, live attenuated vaccine. Okay, in that delta M22, delta G also, delta SH, M22. Okay, this delta M22, in that which model they used? Hemister model they used. Okay, so these are important control strategy. Just once see it and then go in the exam. So I hope guys it's very easy. In very easy way I try to explain you. If you like this video, please like it and share with your friends so that they will also get to know about this hmpv. Okay, so if you have any doubt you can mention in comment. If you want something else, please let me know in the comment box. So thank you for